Greetings everyone, my name is Danial Asyraf bin Salim. I am a final year student from the Bachelor of Science, Biomolecular Science program. I will be presenting my final year project entitled Designing a CRISPR-Cas9 gene knockout system for the GRA2 gene in Toxoplasma gondii. This project focuses on the in silico design of a CRISPR-Cas9 gene knockout system to specifically target the GRA2 gene. To begin, I will provide a brief overview of Toxoplasma gondii and toxoplasmosis, including its significance as a global public health concern. So what is toxoplasmosis? Toxoplasmosis is caused by Toxoplasma gondii, an obligate intracellular protozoan parasite and a major zoonotic disease. It is estimated to infect about 30% of the global population. Although many infections are asymptomatic, toxoplasmosis can cause serious complications in pregnant women, such as congenital abnormalities, and in immunocompromised individuals, including encephalitis and ocular disease. I will then explain how Toxoplasma gondii is transmitted and why it remains a significant public health issue. So, the transmission mainly occurs through ingestion of oocysts from cat feces, or even tissue cysts in undercooked meat, with cats acting as the definitive host. A major public health issue is delayed diagnosis due to the asymptomatic infection, and current treatments mainly target the tachyzoid stage and do not eliminate chronic infections. Following that, I will outline the key problems that motivates this study. The first one is toxoplasmosis remains a global public health concern, and current treatments are limited and may cause toxicity. In addition, the functional role of the GRA2 gene in Toxoplasma gondii is not fully understood due to the limited targeted gene knockout studies, which creates a clear research gap. I will now explain the significance of this study and its potential contributions to the field. This study provides functional insight into GRA2 gene, which is important for parasite survival and host-parasite interactions. It also demonstrates the application of CRISPR-Cas9 as a precise gene knockout tool in Toxoplasma gondii, which serves as a foundational work for future therapeutic or even vaccines development. Next, I will describe the overall methodology and tools used in this study. This study was conducted entirely in silico. The first step is involve the identification of guided RNA target sites using the Toxoplasma gondii PAM sequence obtained from the gene bank. The step 2 is involved designing and scoring the single guided RNAs using the UPA-GDT software. The third step was the primer design using the oligocorp software, and finally the last step, which is by using SnapGene viewer to annotate the single-guided RNA Cas9 complexes. Moving on, I will present the main objectives of this research. The first one, the objectives of the study are to design 3 single-guided RNA sequences for CRISPR-Cas9 to knock out GRA2 gene in Toxoplasma gondii by using UPA-GDT design tool. The second objective is to evaluate all potential on and off targets in silico of the target single-guided RNAs by using scoring-based method. And the last objective is to design primers for single-guided RNAs CRISPR-Cas9 recombinant plasmid validation using the oligocorp software. I will now present the identification of guide RNA sequences targeting the GRA2 gene. For step 1, the complete GRA2 gene sequence was obtained from the gene bank, was used to identify the potential guide RNA target sites. For step 2, UPA-GDT software was used to design and evaluate single-guided RNAs. 3 single-guided RNAs were selected based on high total and efficiency scores, 0 predicting off-target hits, and also the GC contact was between 40 and 60%. These steps ensured the selection of highly specific and efficient single-guide RNAs targeting GRA2 gene. I will then explain the blast analysis performed to assess the specificity of the selected guide RNAs. In this slide, blast analysis was performed as an additional verification step to confirm the single guide RNA specificity. Although UPA-GDT provides scoring and off-target predictions, BLAST is necessary to independently confirm that each single guide RNA aligns only to the intended the GRA2 locus in the Toxoplasma gondii genome. This helps minimize the risk of off-target cleavage, which could affect interpretation of knockout results. Two single-guided RNAs show identical BLAST-aligned sequences because they target overlapping regions of the GRA2 gene, which results in alignment to the same genomic locus. This confirms high on-target specificity rather than a design error. And for the last step, Snap Gene Viewer software was used to annotate the single-guided RNA Cas9 complexes. All of the three single-guided RNAs were successfully inserted into the selected plasmid, which is the FSG671 Cas9 U6 plasmid backbone, confirming the correct in silico plasmid construction. Following that, I will present the primer design results for plasmid validation. OligoCorp software was used to design primers for validation of the recombinant plasmids. All the primers met the criteria shown on the slide, which includes 18 to 24 base pairs length, the melting temperature between 50 and 60 degrees Celsius, and the most important is that there is no hairpin or self-dimer formation. In conclusion, a CRISPR-Cas9 gene knockout system targeting the GRA2 gene was successfully designed in silico using a structured and stepwise approach. All the three single-guided RNAs showed high specificity with no predicted off-target effects, and the design primers were suitable for downstream validation. As recommended for future work, experimental validation should be performed in Toxoplasma gondii tachyzoids. This includes by confirming that the parasites used are tachyzoids rather than bradyzoids, or even contaminants, for example, by detecting stage-specific surface antigens such as SAG1 using immunofluorescence. Following this, functional analysis of the GRA2 knockout can be conducted to better understand its biological role. Thank you.